So we're picking up with video 2 on how does a cell make additional DNA. So a cell has all its own DNA, but we know that during the cell cycle, we make additional cells. And that during which phase of interphase we make a double of our DNA? If you said S phase, you're correct. So in the S phase of interphase, the DNA in a cell is duplicated. The cell makes an exact copy. So how do you go about making that exact copy? How do you duplicate DNA? And the answer is the process of DNA replication. And DNA replication involves the use of complementary strands. So complementary strands deal with the fact that DNA is double-stranded. So when we look at the two strands of DNA, we call them complementary because if this strand on my left has an adenine, then this strand on my right is going to have a thymine. Their base pairs, their nitrogenous bases complement each other. They're complementary. So, base pairs are complementary, blah, blah. So the base pairing rules in effect, A to T, G to C. And DNA consists of a double helix, which we know, twisted complementary strands. And so in order to duplicate our DNA, the very first step of replication is going to be separating the strands. In other words, untwisting the DNA and separating the two strands so that they're available. And each original strand, each original parent strand, once they've separated out, is going to be called a template strand. So here they are together, and then they separate out, and each one is a template. I'm going to use them as a template to make new DNA. So, the first step in replicating DNA is to unwind and separate the helix and produce our template strands. And then we're going to be able to add new DNA to the template strands to produce our duplicate DNA. And we'll just make complementary strands off of each template. And doing that will give us two identical strands of DNA. So how does it happen? Well, the whole process starts at the origin of replication. A large variety of enzymes bind at the origin, one of them being DNA helicase. DNA helicase is an enzyme. We know it's an enzyme because it ends in ASE, right? Helicase. Well, DNA helicase has the job of untwisting and separating the strands of DNA. So, the origin is the start point in the strand of DNA. It can be anywhere in the strand. So, you have something like 10 feet of DNA in a single cell. And so, anywhere in there can be the start point. And so, DNA helicase binds. In fact, you'll get two DNA helicase binding, and they'll move in either direction from the origin. And this makes what's called the replication bubble. And once we've created the replication bubble, DNA polymerases are responsible for adding nucleotides to make new DNA. So here's our parent DNA, and each of these dots is an origin of replication. And the DNA helicases bind, and you can see the arrows going either way, showing which direction these helicases are moving. So we have helicase going either way from every origin. And then this blue is where the DNA polymerases are making new DNA. And so these bubbles continue to stretch out and move continually forward. See, here's the middle one expanding. And as we continue to move, we continue to make new DNA until we eventually get two full duplicate strands of DNA, each made of a template strand, an original parent template strand, and a fresh new strand. Kind of cool. So, here's a picture. This is DNA helicase at the replication fork. And the replication fork is essentially where helicase is unwinding your DNA. So here it is, spreading out your DNA, unwinding and separating it. Then here comes DNA polymerase making new DNA. So the green backbones are the new DNA being constructed by DNA polymerase. And so we get two new strands made up of an old strand, the template or original strand, and the new strand, the green stuff. So pretty easy. So it just continues unwinding all throughout it, making more DNA all throughout it. It just continues on. So I got a couple of videos for you to watch to help you out. Here we go. Ooh, fancy music.
When DNA replicates, its strands are separated by
That's just too annoying. This is DNA helicase, and it's separating the strands. It's one of a variety of enzymes involved in separating the strands. So here come a couple of more enzymes involved in it that you don't need to know the names of, but their job is to keep the strands separated. Well, here's a polymerase. This is a polymerase doing its job, making new DNA. And at the same time, here's the other template strand. And new DNA is also made on the template strand with a slightly more complex process. And because this is 105, you guys are lucky. You don't have to know the details of how DNA is made on the other template strand. But again, we have a DNA polymerase arriving and making new DNA. So we just continually crank out DNA on both parental strands. Here's another DNA polymerase, putting in DNA. And eventually, we create two identical strands of DNA. So, like I said, anything in here that wasn't on the PowerPoint are things you don't need to know. Then here's the other video, which is a little bit more simplified.
DNA replication is defined as the process by which an organism's original DNA is used as a template for the production of a new complementary DNA strand. An enzyme called helicase unwinds the original DNA's double helix, creating a replication fork. Next, an enzyme named DNA polymerase III works down the leading strand and up the lagging strand of the replication fork, synthesizing two new strands of DNA by taking free nucleotides and pairing them with the complementary bases on the original DNA template. The process of DNA replication is described as semi-conservative because the two copies
blah, 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 stuff you don't need to know. So there it is. That's DNA replication. Not too terrible. The links are there in your PowerPoints. And I think we'll stop there because the next concept is something else entirely, even though this wasn't as long as the previous video. You probably like that fact. See you at Transcription Translation, Part 3.